I, on behalf of our department at AIMS Nagar, would like to express our gratitude to have an elite personality amongst us today, our distinguished chief guest, Dr. D.M. Vasudevan, sir. With his gracious virtual presence with us today, this event has become even more glorious, sir. It's my privilege to introduce Dr. Vasudevan, sir. Sir is a stalwart of biochemistry and is very renowned by his textbook Biochemistry, now in 9th edition. I'm sure all of us must have read his book. It is widely used in all medical colleges in India and abroad. Even Spanish editions are also available of this book. Sir has also authored another 4 books on clinical biochemistry. He's a head PG programs and research, Amrita Institute of Medical Sciences, Kochi. Sir has graduated from University of Kerala. He has pursued his post-graduation MD Biochemistry from All India Institute of Medical Sciences, New Delhi. Sir has done his postdoctoral research in Swiss Cancer Research Institute, Lausanne, Switzerland. Sir is a fellow of Royal College of Pathologists. He has profound experience of 50 years in teaching and research in various medical colleges. He has also been a founder, principal, and dean of reputed institutes. Sir has many awards and honors to his credit, and all the awards are very commendable. The few to name are Fellow National Academy of Medical Sciences, FAMS, Dr. B.C. Roy Award for Eminent Medical Teacher, Indian Medical Council. He has received it from a President of India. Sir is a past President of ACBI, Association of Clinical Biochemists of India. Sir has more than 240 papers published in various national and international journals, with more than 4,200 citations to his credit. Some of these are even cited in standard textbooks. Furthermore, Sir has presented more than 200 papers in various conferences, both nationally and internationally. Sir has given invited speech in more than 100 conferences. That is quite a commendable number. And Sir has guided 30 PhD students and Sir is presently guiding 5 PhD students. Sir is a referee and advisor of various committees since 30 years. And he was Vice President of the Indian Immunological Society and is a member of various other societies and university academic bodies. The list goes on and on. To say in short, sir, you have made a society proud with all the great endeavors you have taken over time. We thank you for accepting our invitation and sparing your valuable time with us for this event. We're extremely elated to have you, sir. May I request you to start your guest lecture on advances in molecular genetics? All the students are also online. They have joined, sir, our UG students. So you can please go on.
What is this word? The presentation, is it could not see it here.
Good morning, Vasudevan. Am I audible?
I cannot place my.
Yeah.
See it, sir.
Just say that we can see it.
Yes. Sir, are you able to share the slides?
I can hear. See, I cannot make my PPT. Host the PPT.
Okay.
Sir, can we share screen?
Yeah, perhaps from your side you can put.
Yes.
PPT share कर दो. क्लास शीटिंग शुरू हो गया है.
It is started. Yeah.
Can you see slide?
Yes, sir, it is seen.
Okay, thank you. I am extremely glad to be here with all the dignitaries and the today's topic is certain. Muted, huh? According to me, this is Can you hear me?
Yes, yes, you are audible. Yes, sir. Sir, we can hear you. Your screen is also visible, sir. We can see your PPT.
Okay, thank you.
Yes.
I shall start.
Yes.
Good morning, everybody. Good morning
Yes.
to all dignitaries,
Good morning,
as well as
sir.
participants. These are the major topics that I would like to cover. At
So
first,
all the students are there, you can see them. The students are all listening to your class. Please go on.
can I proceed?
Yes, sir.
Yes, sir, please, sir.
Yes, sir, please. Sir.
These are the major topics, and in the initial few slides, I will describe certain common techniques, and then we shall go a little more higher techniques. And the first one is the DNA hybridization. Before going to any work in the DNA, that we have to isolate DNA. And this is usually done by first from the tissue or blood sample is taken and cell is lysed by TRIS or SDS. And sometimes we use EDTA to remove magnesium because DNase will be present in many of the tissues, and that will decrease, that will remove the DNA, and therefore to remove that we add magnesium. And sometimes we add RNAs so that the unwanted RNA will be removed. And then, of course, protein precipitation and by potassium acetate and phenol chloroform mixture that we isolate the DNA. And sometimes proteinase K is also added. With that, protein and other unnecessary RNAs, etc., are removed. And then finally, DNA is precipitated. And that is by absolute alcohol. Sometimes there will be certain variations in this procedure, but it is usually, these are the usual things. And then finally, DNA is renatured by TRIS EDTA buffer. And there is another way of looking at it, is that removal of isolating the DNA by magnetic beads. That is, you take the sample, lyse it, magnetic beads are added, and that magnetic beads will take the DNA, and the unwanted things will be discarded, the supernatant, elution buffer, and then there will be pure DNA is produced. This is quite simple. If you want to produce so many samples within a short time, this will be useful. In clinical laboratory, most of the time, we use this type of magnetic separation. And now we go to the certain techniques as such, and the DNA hybridization is done by probes. And a probe is defined as a single-stranded piece of DNA labeled, and this labeling can be either radioisotope or non-radioisotopes, and the nucleotide sequence of which is complementary to the target DNA. And this is a basis of the DNA hybridization, that usually we take the DNA from sample. Suppose this is the area it is denatured, single-stranded is produced. A particular gene which we are interested, a corresponding probe is produced. The DNA will be labeled, and this probe, when added, that the DNA, that particular area, wherever there is DNA, that there is the base pair rules, according to the base pair rules, that will be there. And if the DNA is adhered here, that means that particular gene is here, and that you can identify this particular label. And this is one very early picture from myself, DNA hybridization, that there will be different samples are here, and one particular gene we are identifying. In some it is absent, in some it is very less, in some it is very high, like that. We can even quantitate it in this method. Then this is the southern blot technique. This is, see the DNA is isolated, and that is restriction enzymes are added so that there will be, it is made into smaller pieces. These pieces are electrophorized, and then that is transferred to a microcellulose membrane. That is, the gel is here, and over that paper, blotting papers are put, and therefore water will be coming from the tray of the buffer. That buffer will be coming up and up, and along with that, the DNA samples will be blotted into the membrane. In the nitrocellulose membrane, only single-stranded DNA will be adhered. And that is then the radioactivity is added on that. Previously, we were doing radioactivity. Nowadays, the non-radioactive probes are also available. And then the probe is hybridized to restriction fragment. And finally, that is exposed to X-ray film if you are working with radioactivity. If not, the color will be developed, and then we can see these restriction fragments. Wherever that probe is attached, you can identify that. Regarding the northern blotting, the northern blot is to demonstrate, is used to demonstrate a specific RNA. Southern blot is for DNA, northern blot is for RNA. The total RNA is isolated from the cell, and similarly, previous one, everything is same, electrophoresis, and then blotted in the membrane, and RNA DNA hybridization is taking place. And the important thing is that this is used to detect the gene expression. In the previous, that is in the DNA, we can see that that particular gene is available, but whether that is whether that particular gene is working at that particular moment, whether that is expressed, that can be identified by the northern blot technique. And this is the same thing. Sample RNA is extracted, and that is electrophorized. The RNA is separated by size, and the northern blotting is done, and the RNA is fixed to the membrane with, and then that is labeled probes are added and visualized. The Western blot analysis is for proteins. This is not nucleic acid, Western blot. And this technique is very useful to identify against specific protein is present in that tissue, thereby showing the expression of a particular gene. This is the immunoblotting, that the separation in the gel, the samples are taken, that is separated in the gel, and in the blotting tank that is transferred to nitrocellulose, and then in the immunoblotting, immunostaining is done, and then that is developed, and then you can see these marks. Therefore, these are the three blotting techniques that is available. That is southern blot for DNA, northern blot for RNA, western blot for protein. And all the DNA or RNA or proteins are placed on the well and electrophorized, and that is then transferred to nitrocellulose membrane. And the probe is added, either radioactive or non-radioactive, and then you can identify these pieces. The next higher level is in situ hybridization. In situ, that is, see, this is the plate, and over that the cut section is there. That is, the tissue is made in small slices. And then in the tissue, there will be, let us say, gene A, gene B, and gene C. And we want to identify the gene A. In that case, the RNA probe to gene A is added. That of course there will be a labeling. In this particular case, it is DIG, that is digitoidin. That is not important at the moment. It will be labeled. That is a first hybridization, and then second antibody is added, where this antibody against this DNA is added, and that second antibody is labeled with alkaline phosphatase, and in the next level coloring reagent is added, NBTDA. There are so many labels, and blue or purple signal will be the advantage of this method is that you can see the background, that cells or tissues that you can see, and where this particular dye is happening, that we can identify. Therefore, the background is available in the in situ hybridization. It can be done on frozen sections or formalin-fixed tissues, and if the dye is fluorescence, then we can have the fluorescence in situ hybridization, abbreviated as FISH. And it can be other chromogenic substances, then it is called chromogenic in situ hybridization. The major advantage of this is that DNA or mRNA of your interest can be viewed in the context of the tissue morphology. The cells or the tissues can be identified there, and where the particular locality you can identify your particular gene or gene of interest. This is a fluorescence in situ hybridization. You can see that the DNA is taken, probe is added, and that fluorescence will be there. Here you can see the fluorescence that is taking place, and that particular gene of interest that can be identified. The mild modification is chromogenic in situ hybridization, whereas in the surface we have the tissue, the DNA is there. That DNA is biotinylated, and with biotin you can add streptavidin. There will be strong interaction between biotin and streptavidin, and this streptavidin in turn, there will be HRP, that is horseradish peroxidase is present there, and then you add hydrogen peroxide, and therefore that color reagent, there will be nascent oxygen is produced that will oxidize some non-colored substance into a colored substance, and therefore you can identify that particular color. This is something like that. You will get wherever there is that dots, all are the color chromogens, that color is available. And here tissues and cells are also available for your identification. Then in situ hybridization applications are many. For example, gene arrangements in leukemia and lymphoma, diagnosis of B-cell lymphoma, amplification of certain particular genes in breast cancer, HER2 and genes can be amplified. See, these I have taken just 3 numbers. You can make it 300. There are any number of such applications for in situ hybridization, very commonly done in the clinical laboratory. That finishes our DNA hybridization, and then now we go to the polymerase chain reaction. The polymerase chain reaction, or PCR, is nowadays a home word. Everybody knew that thanks to the COVID scenario. That previously the molecular biology techniques were available only in very big centers, and that is not available for common men. But due to this COVID epidemic, now everywhere there are molecular biology laboratory, and therefore PCR is a common feature everywhere. The PCR, this is the basic principle only. The original DNA is there, that is denatured, and single-stranded DNA is produced. This is technically called melting of the DNA, and that is done usually at 95 degrees centigrade. And these 2 DNA strands are there, and you add your primer, DNA primer, that there will be bases or complementary bases will be there, and therefore this DNA primer will attach there. Here also it will attach, and then you cool to 50 degrees centigrade to anneal the primers so that now the primers are fixed there. And then we do the extension that add DNTPs and Taq polymerase. Taq polymerase will withstand the higher temperature of 72 degrees centigrade, and at that level that there will be extension. And now that means that the two strands here, now it becomes 4 strands. This is the step 1, step 2, and step 3. And the primers range from 15 to 30 nucleotides, single-stranded, complementary building blocks of the target tissues. That is this one. And then over that, you extend the DNA. And this is the continuation that this is the denaturation, annealing, and extension. That is 95 degrees centigrade, 50 degrees centigrade, and 75 degrees centigrade. And then the same thing can be repeated. That is the second cycle. And by the end of the second cycle, there are 8 pieces of the DNA. And then the third cycle comes, and then there will be 4. And by nth cycle, you will get 2 to the power of n molecules. That is, millions of times the DNA is amplified. That is why it is called PCR, the polymerase chain reaction and amplification of DNA. These are usually done for the diagnosis of bacterial and viral diseases. In the early phases of tuberculosis, the sputum may contain only very few tubercle bacilli, and therefore staining may be negative, and therefore you will miss the diagnosis. But PCR could detect even one bacillus present in the specimen. The specific nucleotide sequences of the bacilli are amplified by PCR and then detected by the blot analysis. Therefore, that is very common. And I am taking the tuberculosis only just an example. Any number of such infections can be identified. Medical legal cases, PCR allows the DNA of a single cell or in a hair follicle to be analyzed. And again, restriction analysis of DNA is done. And then the pattern is then compared with the DNA sample obtained from various suspects. And the culprit sample will perfectly match with the PCR amplified sample. Diagnosis of genetic diseases. PCR technology has been widely used to amplify the gene segments that contain known mutations of the diagnosis of inherited diseases, such as sickle cell anemia, thalassemia, cystic fibrosis, etc., etc. Previously, the genetic disorders, difficult to identify that particular gene. Now, with the PCR technique, you can amplify that, and then therefore you can identify. Of course, parental diagnosis of inherited diseases. Cells can be obtained from amniocentesis, and these are very few numbers only, and therefore you need a minor extension of this technique, that is the reverse transcriptase PCR. That is the PCR, before the PCR, we do the reverse transcription. This is the RFLP viruses. The typical example is your COVID virus, SARS virus, and that the RNA is there. That RNA is made into reverse transcription. The primer is added, and then the transcriptase, reverse transcriptase enzyme is added, and that RNA part is made into complementary DNA, and then PCR, by PCR technique, this complementary DNA will be amplified many times, and after that you can do the blotting. Reverse transcriptase PCR, instead of Taq polymerase, usually TH polymerase from Thermophilus may be used. This enzyme has both DNA polymerase and reverse transcriptase activities at 95 degrees centigrade. RNA is there will be amplification. And in ordinary PCR, DNA is detected. That DNA could be from a living or non-living organism, but in reverse PCR, we are detecting the mRNA, and that means it is derived from a living organism. That is another advantage. Moreover, as I have already told, RNA viruses, you have to do the reverse transcription before the PCR. Another modification is real-time PCR, that is otherwise called quantitative PCR. By this method, quantitation of the number of viruses present in the sample can be calculated. And that is important clinically. See, for example, you want to treat HIV and the viral load has to be determined and your treatment modality will be changed depending on the number of the viral load. And therefore, in this, similarly, an HBV, that is hepatitis B virus also, the number of particles, how much is the treatment depending on that. And how to do that? See, this is before the threshold value or threshold penetration. See, in this area, we are doing the cycling, that is and the DNA sample is being increased. And at this level, there is a increase exponentially. And here, up to this line, below this line, that you cannot identify or you cannot detect because this is below the threshold. And after that, you can get the number of DNA is available and therefore you can detect it. And therefore this is usually say 30 times you are cycled and getting this available of the DNA. Whereas suppose your original sample is very low, then more 35, 40 or RT-PCR or qPCR. RT-PCR is also told that reverse transcriptase PCR. So it is better as qPCR, that is quantitative PCR. See, you can take, let us say, 0.1 nanogram, 0.1 nanogram, 1 nanogram, 10, 100 like that initially, and then do the test, and you will get different these graphs. And along with that, the unknown sample is put, and therefore from that you can see what is the quantity of the unknown. And that is the way by which the quantitation is being done. How many PCR cycles are done like that. And another modification is multiplex PCR, that more than one target sequence can be amplified by using multiple primers. That is the primer set. Instead of one primer, you take, let us say in this particular case, A, B, C, D. And therefore 4 primers are added and therefore 4 areas of DNA will be amplified together. And therefore 4 times amplification you will get. In fact, you can go very high quantity of amplification, and therefore that is a quicker method for multiplication. Thus, we have finished this polymerase chain reaction. Next is the lamp technique. Pardon me? This is the lamp technique. Please see that the lamp technique is quite new to India. Of course, lamp technique was there for the last 10 or 15 years in the world. But in the Indian market, this has come only recently. And that is, therefore, we can say that this is a recent advance. The LAMP stands for loop-mediated isothermal amplification. And usually in this is a very common name nowadays, RT lamp, and because of the COVID infection, the RT means reverse transcription, and then lamp, that is the loop mediated isothermal amplification. We shall take a quick look of how this is being done. And the RNA isolation is done. This is in the RT-PCR, RNA isolation is done. And that is RNA is extracted by chloroform isopropyl alcohol. Add 50 microliter of magnetic beads. Nucleic acid attaches to the nanoparticle and mix and keep the magnetic stand for 2 minutes. Wash. RNA is isolated, and to that primer and reverse transcriptase is added, and cDNA is synthesized. After that, it is the routine PCR, which I have already described. What are the primers used? The major difference with LAMP technique is that these primers, 4 different primers are used. One is forward inner primer, FIP. Second one is forward outer primer, FOP. Third is backward inner primer, BIP. And fourth is backward outer primer, or BOP. I shall give all the details. See, this is our original, let us say, our original DNA. The two DNA strands are there. We separate that, the one strand is here. And in that, the forward inner primer is added. That is this F2 here, in the F2 ring is added. And this is the so-called forward inner primer region, the whole area. And this F2 is added. That will attach the, and to that DNA polymerase is added. The enzyme is working on, and from 3' to 5' that there is the DNA being synthesized. In this step 2, the DNA polymerase is added, and therefore that will complete that DNA strand from here onwards. And then in the step 3, F3 primer is added. This is the F3 primer. This dark block is the F3. And that is added, which will have complementary sequences in this in the original DNA. And that is the forward outer primer. This is outer primer. The previous one is the inner primer. And double strand is formed from DNA strand synthesized. Therefore, this is the DNA newly synthesized DNA strand. In step 5, that we make the strand displaced. The newly synthesized strand is displaced, and this is the newly synthesized strand. You can see that this F1 and F1C, these regions will have complementary sequences, and therefore this end, a loop is formed. Yeah, this is the loop is formed. That is why the technique is called loop isothermal reaction. Then in the step 6, that the other side, that is the backward inner primer is added. This is the backward inner primer. Similarly, that is attached here, and to that you are adding the polymerase enzyme, and therefore enzyme will work it here. And then B3 is added, the backward outer primer is added, and finally, there also the double strand is produced. Then step 8, that this one is now, you can see both ends. This is the DNA that is coming, and that lamp method, this in the both ends, you will get a loop. And that is why this now onwards, this is the amplification. From here onwards, it is the amplification cycle is going on. Now these loops will act as the primers, and from here, both sides, the enzyme can work now. That is stopped here. This is the original DNA strand. Over that, a new strand is produced, and over that, that is another loop. Again, that is going on. Therefore, this will go on and like that. And the other strand also, it is going on like that. Therefore, within few seconds or within few minutes, actually, you will get an enormous number of multiplication, enormous number of amplification. The previous few slides are condensed here. That is the double strand is there. You are adding this forward inner primer, forward outer primer, etc. And here the other end also the same thing is added. And therefore DNA synthesis is here, DNA synthesis is here. Therefore, in both way, that there will be a loop is produced, and that loop will act in the next sequence reaction, next polymerase chain reaction. Therefore, these loops are added, and because of that, there will be any number of the multiplication. Now, this is called loop mediated. I have told why it is called loop, and this is why it is called isothermal, because in the normal PCR, which I have told, that there is a cycle. That first we put it at 95 degree, and then cooling at 50 degree, and then 72 degree. And this is a cycle. Whereas here it is isothermal. The RNA LAMP test, that it is RNA is extracted, a reverse transcriptase and cDNA is produced, and the LAMP amplification is there. And finally, you usually add a dye, usually a fluorescent dye is added, and therefore you will get a negative or positive test like that. A quick reaction is available. Then this is a comparison between PCR and the new LAMP technology. In the PCR, it is a variable temperature, 95 degree, 50 degree, and then 72 degree. Whereas here isothermal, it is only the routine, 37 degrees centigrade, you will do all the test. And therefore that is a great advantage, because here you need a thermal cycler in the usual PCR, whereas here there is no need. Therefore the equipment necessity will be reduced. Whereas PCR you need minimum 6 hours, usually 8 hours, and usually you will get the result only after 24 hours. Whereas here you will get the result of the PCR work will be done within 20 minutes, and the result will be getting within 2 hours. That is the greatest advantage. Here your yield is comparatively low. In the new technique, yield is very high, because as I have told that the multiplication is far robust. Here the amplification is specifically is lower than that of LAMP. Amplification specificity is higher because we are using minimum 4 oligonucleotides, whereas here it is only 2. And here the PCR need pure DNA samples, whereas this one you could use a crude DNA sample. And therefore for routine clinical work, the LAMP will be better utilized. And PCR costly detector is, and here the simple detector is there. And therefore in all these, that the actual cost will be reduced, especially the infrastructure cost will be greatly reduced. And therefore for each LAMP assessment, there should be, this is the disadvantage. That is for LAMP assessment, the specific designing of the target DNA is necessary. The probe designing is very important. Of course, that how to design it, etc. You will get the demonstration, etc. later in the evening. And therefore, however, there are softwares are now available for primary exploration. Of course, that the initial standardization will be difficult, but when it is in a kit form, it is very easy to do. Even small laboratories can do the test, and that is why the molecular biology laboratory techniques are now coming up in a great scale in India. Then for applications of LAMP, it's a rapid diagnosis for viral, bacterial, parasitic diseases, genetically modified ingredients, tumor detection, etc., etc. Therefore, there is anything that the PCR, the old PCR, can be modified into LAMP technique with quicker results. That is the advantage of this LAMP technique. Therefore, that LAMP technique is also over. Next, we go to restriction fragment length polymorphism, single nucleotide polymorphism, etc. Restriction enzymes you are familiar with, that let us say an enzyme like ECOR1 that will cut the DNA in such a way that there will be staggered digestion and or sticky ends are produced. That is, and any number of restriction enzymes are 600 to 700 restriction enzymes are available with the specificities. And this RFLP means restriction fragment length polymorphism. You take the blood sample, isolate WBCs, and this can be taken by any tissue you can do like that, and then DNA of the particular small segments of DNA is taken, and that is further digested by restriction enzymes, and fragments are separated by electrophoresis and southern blotting. This is, I have already described the southern blotting. This is the same, more or less same thing from here onwards. The labeled DNA probe is hybridized. There will be autoradiography and DNA profile is there. This profile you can identify that the particular gene changes in the mutation in the gene can be identified by this method. I shall come to that. An example is sickle cell anemia. The sickle cell anemia, you can see that, see, this is the sickle cell gene and this is the normal globin gene. And this particular MST2, that particular enzyme will cut this region. Therefore, only one piece of the gene will be available. Whereas in the normal circumstances, normal globin gene, this MST2 will cut here also. Therefore, you will get two pieces. And then you do the southern blotting as usual, the electrophoresis, hybridization, etc. And finally, you will get a result like that, that in the case of normal, you will get two lines, whereas in the sickle cell anemia, that you will get only one single band. Therefore, this is a very quick method to identify the genetic diseases like that. That is why it is called the restriction for restriction segment polymorphism. Same way, you can identify a particular person. Each of us are different. One person is different from another person with regard to the DNA. Blood sample is taken, DNA is extracted, cut by restriction enzyme, and these DNA fragments are separated by electrophoresis. Southern blotting is done. Radioactive activity is DNA probe is hybridized, washed, radioactivity bound to DNA to the membrane, and X-ray film is placed, and then film is developed. And then the pattern from each person can be tested. And that it is in the conventional DNA fingerprinting, there will be hypervariable and repetitive sequences are taken as the probes. That is mini satellite or microsatellite DNA. Anyway, there will be that particular DNA samples are taken as the probes. And this is one example. Let us say this is a crime scene is taken, and in the crime scene, the culprit's DNA sample is like that. You will get these lines. Now these are 1 and 2 and 3 suspects. You don't know who is the actual killer, and you can see that here this particular there is no identity, whereas here this particular line there is no corresponding identity here. Similarly here, whereas the sample from now the suspect number 2 that exactly matches from the sample that is taken from the crime scene. That means that this particular person is the culprit. That is why the way by which we identify this crime. Now, why it is possible? Because only one person, every 10 million persons will have a particular DNA profile. And it is therefore extremely unlikely that you will share the same profile as someone else. Let us say this paternity test. In this particular example, who among the children are real sons and daughters who are adopted? This is the mother's sample is like that. This father's sample is like that. And this in daughter 1, you can see this line is corresponding here. This line corresponds here. This line corresponds here, corresponding here. And similarly, this one also, that there will be either father or mother, that particular line will be available in the daughter and this particular son. Whereas this particular is also son, but none of these lines can have any lines with regard in the mother or father. Therefore, you can immediately say that this is the adopted son. And in that way, paternity tests can be done That leads us to single nucleotide polymorphism or SNP. SNP is usually pronounced as SNP, and SNPs are substrates. One single base is changed. And SNP occur every 300 bases, so approximately 10 million these differences will be there, and that is why one person is different from another person with regard to the DNA analysis. These differences can account for the differences in disease susceptibility, drug metabolism, response to environmental factors, and response to different drugs, etc. And therefore, the single nucleotide polymorphism, that is SNP analysis, has become a great tool in the molecular biology. Now, this is exemplified here. See, you can see that here T and A, here that becomes another, and that is a mutation is taken place. This is a single point mutation, that is why that is called a single nucleotide mutation, yeah, SNP. There is also another term called microsatellite markers. And microsatellite markers are also called single tandem repeat, simple sequence repeat, simple sequence length polymorphism. And that there will be such a number of repeats varies from 5 to 30 or something like that, sometimes even more. Sometimes even hundreds are identified, and there will be highly polymorphic, and it is seen throughout the genome. This is the schematic representation of the difference between SNP and microsatellite variation. I have already described SNP, that is here T is that nucleotide is changed to A here. That means a mutation is taken place. But that mutation is with regard to only one nucleotide. And that is why it is called single nucleotide polymorphism. Whereas microsatellites is this one. Let us say here CTA. That CTA is repeated 5 times here. Whereas in another person, CTA is repeated 6 times. And another person, CTA is repeated 7 times. And that is why it is called microsatellite repetitions. That is the difference between SNP and microsatellite. Then SNP may or may not be associated with a disease condition. In this following example, it leads to hemophilia. Now let us say this is the wild type factor 9 gene. And there is a variation has taken place, the C to T where mutation is taken place. And what happened to the protein that is produced in the wild type, that is translated as arginine, is CGA, that is arginine, whereas TGA is a non-stop signal, and therefore the protein will not go further, protein will be cut there. No more synthesis. And therefore, the normal protein is not produced, only a truncated protein is produced. And that leads to hemophilia, and therefore that particular gene product is not available, factor 9 is not available, that becomes hemophilia B. Now, how can you identify SNPs? Of course, theoretically, you can identify that by gene analysis, that is the nucleotide sequence you can do, but that is very time consuming and also money consuming. Whereas a quick response can be done by denaturing high performance liquid chromatography, DHPLC. This is used to detect single nucleotide substitutions or this is used to detect single nucleotide substitutions or small insertions or deletions. That is only 1 or maximum 2 nucleotide chains that can be identified by this method. Can identify DNA fragments that contain sequence variations, but this DNA sequencing is required to confirm the precise nature of the mutation. With regard to DHPLC, you can tell that there is a mutation. But what particular is the new mutation that you have to do the DNA sequencing later, if necessary. And generally, DHPLC is performed under partially denaturing conditions. And it has been demonstrated that for shorter fragments like less than 30 nucleotides, it is possible to separate all oligonucleotides differing by a single base. This will be a feature like that. See, suppose fully paired, all the nucleotides are paired, and therefore you will get something like this. Whereas if there is mismatch, then you will get that particular area, there will be a bubbling that cannot, the correct DNA structure is not formed here. And therefore, alteration of structure because of that one particular DNA the sequence is altered. And therefore, if you take the normal sample, there will be only one peak, fully paired DNA. It is sometimes called homoduplex, whereas in the mismatched, you will get a two. That is called a heteroduplex. And then immediately you can say that this particular gene, there is a mismatch. That means there is a mutation. What is that particular mutation? We have to go for the DNA, the next level, and before going to DNA sequencing, I shall go to DNA microarray. Microarrays have come in great way in the molecular biology in recently for the diagnosis of diseases as well as for diagnosis of the infections. Suppose we want 10,000 genes are to be quantitated. The answer is microarray. This is happening in most of the newer techniques that unless you diagnose, you go for various genes, and that will take a lot of time in the previous generation. But nowadays it is possible. It is a high throughput technology that allows detection of thousands of genes simultaneously. The principle, the probes are fixed on a solid support like a glass slide. The print tip is a needle-like steel pin that takes up a few nanoliters of fluid by capillary action, and then there is robotic printing. It is simple in theory, very complicated in actual practice. The machine is something like that you will have a plate. In that you are put all the, let us say, thousands of samples, thousands of different genes, small genes are put there, and the printing head will take up a few nanoliters of the liquid and poured over to slides, and of course washing, etc. Therefore, this will take a few hours for the printing. Generally speaking, 10,000 gene array is made within 13 hours of such printing. So, this is the DNA array. That this is the chip or as such. The actual chip size will be this one. This is the actual chip size. And this will be an enlarged form of that. In this particular case, it has about 1,000 locations. And to that is again magnified here. And in all this, you are adding certain particular DNA sequence. That is, each gene can be made here. Millions of DNA strands build up on each locations. And therefore, this is the tagged probe is already there, that is tagged. And to that, your patient sample is added. That is why this is made. The technique in the simplest form is like that. RNA is isolated. Let us say this is from a normal person, and this is from the diseased person, let us say a cancer patient. The cDNA is generated by reverse transcriptase, and therefore this label is then probed with fluorescence in both one, and then hybridized to array. This array, there are already in that plate, each location will contain one particular gene. Let us say thousands of genes are now there. And in all these wells, you are adding either the normal gene or the cancer patient's genes. And then that this will come like that there will be, let us say, these black ones are gene not active. And this is green one, gene ones are normal active. And the red ones, gene active in the disease. And in the yellow one, genes inactive in both normal as well as in cancer cases. So for that array, you can immediately, of course, this is, of course, finally it is a computer analysis. But anyway, this is a theory of microarray techniques. Therefore, thousands of samples can be, thousands of genes can be identified, can be whether that particular gene is amplified or reduced or in normal way, that can be identified by the microarray technique. Nowadays, in the cancer diagnosis and cancer chemotherapy, which particular gene is increased, then that particular modality can be, treatment can be given. Microassay is for diagnosis of fetal chromosomal aberrations. Let us see how it works. This is the patient. Let the patients, you are adding green label, reference that is the normal, red label is added, and you hybridize, and then the analysis will come like that. The patient, this is the patient, and this is the control. And patient and control both having 2 copies. That means that chromosomes are 1:1 ratio, correct. Whereas here, patient has 3 copies instead of normal 2 copies, and therefore there is an extra chromosome is available. Whereas here, chromosome only 1 copy in the patient, and the normal 2 copies. That means that one chromosome is deleted here. Therefore, that is the fetal chromosomal aberrations. This is a quick diagnosis. Within 1 hour or something like that, you can get the idea of such chromosomal aberrations. That is the great advantage. And fields of application of microarrays, so many: tumor diagnosis, infectious disease diagnosis, fetal chromosomal aberrations, allergic diagnosis. See, with regard to infectious disease diagnosis, I would like 2 minutes on that. Let us say a patient is coming. He has a fever. We don't know which microorganism is the culprit. It can be a bacteria, it can be a virus, etc. Now, theoretically, at least, now you can have here in a plate, there can be 1,000, all the pathogenic bacteria and viruses can be put in that. And you just put the patient's serum or patient's sample, then immediately you will get the diagnosis. Within 1 hour or 2 hours, the diagnosis, what particular microorganism is there, that can be identified. Of course, the early stages of such microorganisms diagnosis are already available in India, and very soon the whole lot of infectious diseases can be identified by molecular disease, by molecular. Previously, the molecular biology techniques were very difficult, and only very big centers can do that. Nowadays, it is very simple methods are already available, and therefore even smaller laboratories can do that, and therefore more and more molecular biology techniques are being used nowadays. DNA microarray is also over. Next, we shall go to DNA sequencing. DNA sequencing is for, as I have already told, that mutations can be identified, which particular mutation specifically you can identify, and order of nucleotides in a DNA molecule. In the Sanger sequencing method or chain termination method is the usual method that we adopt in the laboratories. And this method uses the synthetic nucleotide which lack the 3' hydroxyl group. And therefore, these are called dideoxynucleotides, DDNTPs, or D-deoxy. Anyway, that the third third 3' position hydroxyl group that the phosphoric acid is not there, and therefore they are unable to form the next phosphodiester bond, and therefore chain elongation will stop there. Wherever you get this DNTP, that chain will be lost there. Chain elongation will be lost there, and therefore a smaller fragment is produced. That is schematically represented here. This is our template, the normal DNA. You add the primer, necessary corresponding primer is added, and then all the normal NTPs, that is the ATGC, all these with the NTPs are added, and to that mixture you are adding DDATP, DDGTP, DDCTP, and DDTTP also, and you start the polymerase reaction, and that means randomly these unwanted things will enter. See, the polymerase reaction is going on, newer nucleotides are added one by one. Out of that, erratically, that is the one DD will come here, and then the elongation is stopped here. And then the next minute, there will be another thing is synthesized, but that length will be different. Therefore, let us say that you have 10 nucleotides here. All the 10 nucleotides will come like that. That there will be this much length, another length like that. And therefore, a polymeric form of different forms of DNA is newly synthesized. And now you can see that the molecular weight of this and this will be different. And therefore, you can immediately do a electrophoresis, and there all these lines will come like that. Now, from this, you can decipher what is there. See, for example, in the first here, DDTTP is added, and therefore that is a red signal. Therefore, here, this is the smallest segment, and therefore that will, the smallest fragment will go a longer way in the electrophoresis. Therefore, this number 1, that is red. The second one here, that is also red. Third one is yellow. Yellow means GTP. And like that, you can get, immediately you will get what is the sequence. And that is the basic principle of DNA analysis, the Sanger sequence analysis. It is very easy to tell, but practically difficult. If you remember the Human Genome Project, it took nearly 10 years to complete the whole human genome to be analyzed by this method. And you cannot wait in a clinical laboratory for that much. And therefore, there should be quicker methods should be available. The same principle I have elaborated here. This is from my textbook. That is, these lines you can see immediately what is that particular nucleotide, and then the length of the sequence can be identified immediately. As I have told that the Sanger's technique is very cumbersome, it will take a longer time. And therefore, there should be other methods that is now come, that is called the next generation sequencing. It has already come in India. There are a lot, almost all big laboratories will have the new generation sequencing available, while the Sanger method is only sequencing a single DNA fragment at a time. NGS is sequencing millions of fragments simultaneously per run. And this process then translates into sequencing hundreds or thousands of genes at one time. It is otherwise called a massive parallel sequencing. In very nutshell, it is like that. The biological sample is taken, genomic DNA is isolated, and that is fragmented by DNAs, and then adapter ligation is added, that is, and then each of these are sequenced parallelly. Suppose there are 100 small nucleotides are available, 100 different sequencing is going on simultaneously in the machine. And then finally, it is a computer alignment which comes first, which molecule comes first, etc. And therefore genetic coding can be done. Uses of NGS in clinical practice for clinical genetics, microbiological identification, oncology, personalized therapy. These are all that is now coming up in a great manner in Indian scenario. Now, and still easier method, quick method has come, that is called a nanopore sequencing. It is the Oxford company, Oxford nanopore sequencing devices that use a flow of cells that contain an array of tiny holes. Through the holes, the DNA, single-stranded DNA is passing through. And when particular nucleotide is passing through that pore, that there will be a change in the electrical neutrality, and therefore that can be identified. That current, the molecule passes through the nanopore, the current is disrupted, and therefore that will be identified. Using a nanopore sequencing, single molecule of DNA or RNA can be sequenced without the need for PCR amplification. And complex formed between R-hemolysin and stranded DNA with the linkage through the oligo. That is what he is here. This is the enzyme, and that particular enzyme, a protein, a particular alpha hemolysin protein, creates a molecular nanopore, and through that this DNA is passing through, and the flow of ions are here, and when that particular one DNA, that nucleotide is passing through that there will be the current of that particular area will be disrupted, and that is analyzed by the machine. And the advantage of this one is that you can identify the DNA sequence within 1 hour or something like that. Therefore, that is the quick method. The disadvantage is that specificity is less, it is only 90% or something like that. Therefore, errors can be there. Otherwise, this can be a very easy method that is now coming up. And another advantage is that the cost of the machine is also less. And therefore, these are the certain advantages of this nanopore sequencing. And we are going for the last technique, recombinant DNA technology. And this is, of course, well known. The restriction enzymes will, as I have already told, that it will cut in specific areas, and then there will be sticky ends are available, and you do in the plasmid in preparation. Plasmid is cut with echo R1. Similarly, the gene, human gene, is also cut by the same echo R1, and therefore that will have the sticky ends here. This plasmid also will have the sticky ends. And now, if you anneal or join by DNA ligase, that recombinant DNA is there. It is easy to tell, but there will be certain difficult technical details. I don't want to go in greater detail at the moment. Now, it is transfected, that is in the host E. coli cells will, and the plasmic vectors are incubated in hypertonic medium containing calcium. Within few minutes, that there will be host cells are allowed to grow. Therefore, their plasmids are entering into the host cell, and they are in the agar platelet is there. And only, of course, 5% of bacterial colony is containing the desired vector. Therefore, there will be so many bacterial colony that we have to identify, and that is by this transfection. This plasmid, this particular plasmid will have APR, that is an ampicillin resistance gene, tetracycline resistance gene, and chloramphenicol resistance gene. And if you add a particular insert, that is a particular human genome is added here, that insert, and therefore this CMR is broken. And therefore, now what happens? The chloramphenicol, that resistance is gone, and therefore this chloramphenicol can act on this particular bacteria. Therefore, this transfection colony can be identified. Let us say here, the E. coli bacteria is growing in ordinary medium, and therefore many colonies are there. But here you are adding ampicillin and tetracycline, and therefore only few colonies. And this is replica plated, that is with chloramphenicol, where chloramphenicol sensitive colony is marked as red. And therefore this particular colony from here, that is the corresponding that particular colony here, is taken, looped out, and that colony will be expanded. That particular colony will be killed here. And therefore, this particular colony is the colony that we want. And that is human mRNA is taken, cDNA copy is produced, and plasmid vector is cleaved by restriction endonuclease, that is transfected into host bacteria, and there is selection of bacteria carrying the human genome by antibiotic sensitivity, and then that particular clone is selected, amplified, and from this cell culture, we can isolate the proteins. Theoretically, very simple. And let us say, if you wanted to produce human insulin, that the DNA from the pancreas is taken, mRNA from pancreas is taken, mammalian proinsulin mRNA is taken, reverse transcriptase, and that this is the proinsulin cDNA is produced, that is joined in the plasmid, and the gene of particular interest, the proinsulin is here, that is infected the E. coli, and E. coli now transformed bacteria will produce the proinsulin, and that can be harvested. That is the way by which we go for the genetic transmission. Recombinant proteins are nowadays available plenty. Hundreds of human proteins are now available, not even hundreds, may come to thousands nowadays. By recombinant technology, insulin, interleukins, interferons, antihemophilic, globulin, growth hormone, etc. In the previous era, when growth hormone is required 1 unit, that needs the pituitary of 1,000 cadavers. But and therefore it is practically impossible to get human growth hormone. Nowadays, it is plenty available. The availability, that is the importance of recombinant proteins. Similarly, let us say hepatitis B surface antigen. It is a tedious procedure to isolate the antigen from the contaminated plasma, whereas here you can produce it by recombinant proteins, and therefore there will be no problem of contamination here. With that, I remain. Thank you very much, and I have taken 1 minute, 5 minutes more. Thank you.
Thank you, sir. Thank you for enlightening us with your knowledge in the arena of molecular genetics. Hats off to your enthusiasm and energy, sir. The students are really fortunate to hear you out. Our UG's first, the second batch of UG students. Thanks a ton for your pearls of inspiration, sir.
Okay, thank you. Welcome.
HOD madam.
No, thank you very much for all the listening.
Yeah. Sir, we request you to accept virtual memento, sir.
So we should send it to you, and I would like, please accept this. Please come.
Thank you very much. Thank you. Thank you. It is wonderful. It is. I am greatly honored
This is for you from AIMS Bibinagar.
Oh.
And I would like to tell you that I can see another college in Pune. All the UG students are also attending your lecture.
Oh, thank you.
Who have reached out to a lot of students, inspiring them. And thank you very much. And at this age, the way you took us through the journey of molecular biology, right from basics to the latest of nanopore, it was really amazing. Thank you very much.
I am honored.
Thank you, sir.
Thank you, sir. Sir, we had a question from the audience.
Oh.
One of the students was asking what is the difference between SNP and mutation?
No, SNP is a mutation, but the mutation can be many types. Mutation means alteration in DNA sequence. And that can be at one particular point, that is one nucleotide is altered, then that is called SNP. If there are many nucleotides are changed, then that is for usual mutations. Mutation means usually many alterations will be there.
Yes. Sir, thank you for that answer, sir. There's one more question, sir.
Yeah, please.
What's the difference between dPCR and qPCR?
Pardon me once again.
What is the difference between dPCR and qPCR?
qPCR is actually the quantitative PCR, which I have described in greater detail. That is, see in certain cases, we need to get the knowledge of how much DNA is available. Quantitation of DNA is needed. I have told that, see, for example, HIV infection. HIV, the treatment, the dose of the drug as well as the choice of the drug will depend on the load of the HIV in that particular patient. And therefore, quantitation is required. That is why we make the quantitative PCR. That is, known quantities of DNA is taken, amplified, and in that you will get a graph, and correspondingly you add your patient's serum or patient's sample, and then you will get a particular type of type, and that will from that graph you can identify the quantitation. Yes. Any next question?
There's one more question, sir.
Yes.
Is that PCR should be done before DNA sequence?